Has more than 15 publications in various national and international journals, and he has filed 7 patents, and he also has a book chapter, though his credentials are more. But with this small brief introduction, I hand over to Dr. Gyan Prakash Modi, and his topic for today is drug engineering for pathogens.
Okay, good afternoon. And, I'm again very much thankful for the organizers to give me this opportunity to deliver a talk on in this FDP program. It's always, very helpful to share the science, and then people can discuss on this, and which can be further, can be utilized for the purpose of society. So I just like to get an
Can I?
idea. I assume my voice is coming, and I am audible, as well as the screen is also coming there. Am I correct?
Yes, but I was just saying adjust your camera because we cannot see the whole of your face. We are just able to see your specs and
Oh, really?
yeah. So just adjust your camera such that we'll be able to see your.
Okay. Is
Yes,
it
this is
coming?
better. Yeah, this is perfect.
Okay, sounds
This
good.
is yeah. Thanks.
So yeah, fine. Thanks for correcting me. So as I said that it's always important to share the science. So then it can be utilized. So it always multiplies when you share the things. That's the old saying. We all are aware about it. So I just like to know about the participants. Can you just give me a brief idea, the background of the participants? Are they aware about, about their background? Are they, they're doing some kind of research, or the faculty members? Can you just give me a brief background about the participant?
All pharmacy faculty, and they're all
Uh,
with less than 10 years experience, teaching
okay,
experience, and they're from various departments like pharmaceutics, and some must be from pharmacology. Also, they're
okay,
from all over. Like we have people from Goa, Maharashtra, Karnataka.
okay,
Those who have
okay.
enrolled here. Yes.
Okay, so with this, I will start my talk. You can always stop me at a point when you feel. Um, um, so when I was thinking what I should discuss about, so this topic I thought about is drug engineering for pathogens. The reason I thought to deliver is given the current pandemic situation. And I have I did my postdoctoral training at Brandeis University, located in Waltham, Boston, with Liz Hedstrom and Greg Cuny. So during that time, I worked on the antibiotic development. So it's a very important thing that how you can think of engineering a drug for pathogens, because pathogens are very different. Right? COVID probably is the most common thing for all of us to give an example of pathogen. So the reason is that the pathogens, when you talk about, you are going to deal different kind of the barrier to kill these pathogens. So I will start with one of the simple pathogen, which is called as a Cryptosporidium Parvum. I will talk about the how you can engineer the drug against Cryptosporidium, which is a parasite. And from there, the next I will enter into the designing or engineering the drug for bacteria. And there I will try to connect with some other kind of the pathogens. So Cryptosporidium parvum, I'm not sure you might have heard about this parasite or not. आप में से किसी ने सुना है नाम या नहीं? I do not know. But let me just explain you in briefly. Cryptosporidium parvum is basically a parasite. I'm just trying to put in a simple way so you can understand it. Is a epicomplex in detail, is a epicomplex parasite, and is basically mainly used to sit in the intestinal epithelium and do the infection at the intestinal epithelial site. So one of the major reason of diarrhea, malnutrition, and specifically in kids, in the children is a one of the severe cause of death as well, because of the nature of the diarrhea it causes. It can be life threatening, specifically in immunocompromised people, and it can also be utilized as a biowarfare agent. I am sure that you know about biowarfare agent. Biowarfare agents are such which can be utilized as a biological weapons. Maybe one country can throw the one bottle full of the Cryptosporidium parvum to the other country, and then it can cause the infection to the other country. Unfortunately, there is no drug so far, and no vaccine as well, for Cryptosporidium parvum, because of the nature of this parasite is a really severe. It can cause the severe problem in children. So outbreak, like we are recently looking at pandemic outbreak. Similarly, this outbreak happened in 1993 in Milwaukee, is in the US, where approximately 4 lakh cases occurred because of this Cryptosporidium parvum. And in the US, in summer 2014, again, because of this, raw milk is one of the major source, because of this, it comes through the cow and other animals, and where the cleaning and those things probably may not be very good. So this can enter into the milk, and then it can go into enter into the human, and then eventually once it enters into the intestine, it's going to propagate, and eventually can cause the problem. So if you look at the the causes of the death in kids, specifically the below 5 years. So rotaviruses, probably you might have heard about. Vaccination is a common thing which usually we take our kids for vaccination against the rotaviruses. So vaccines are available against rotavirus, but rotavirus is one of the major cause of the death due to diarrhea in kids. You can see that diarrhea is major cause, around 15%. Second is the pneumonia. I mean, another is the pneumonia, which is another cause of the death in kids below the 5 year, and malaria. And there are other factors as well which contribute into this. Rotavirus is it causes around 7 deaths per 100 children per year. Cryptosporidium around 3.4. Another parasites are also there, which are also responsible for the for death of kids in due to, specifically due to these are all the pathogens, the pathogen induced diseases. So what happens to Cryptosporidium parvum? As I said, that the infection can happen through the milk, and when you take when there are other sources as well. One of the typical example I took is can be through the raw milk. Once it enters into the intestine, is going to propagate there. So this is the structure of the of Cryptosporidium parvum parasite. You can see the shape of this parasite is pretty unique. It sits into the intestine villi, and then it used to replicate over there and keep multiplying, and eventually it will undergo the meiosis process, and it gets another gets released out of the intestine and transmitted to the other host. So this that whole replication process happens in the intestine valley. So once it it will have enough number, because some of the infections, they are self-limiting as well. Probably you might have heard about self-limiting infections, where infection will happen, but it is not going to cause the severe infection. So it is self-limiting. It itself will not going to multiply in thousands or or more before it can produce the symptoms. So Cryptosporidium parvum keep multiplying, but sometimes it is self-limiting as well, specifically in the elderly people, because of their immunity is more stronger. So now if you look at this Cryptosporidium parvum parasite, this is the again intestinal villi where this parasite is sitting. So this green you see is light green is a parasite, and this is the our intestine. So this is the host, this is the Cryptosporidium parvum. What we look over here, So from intestine, you can see that we have a complete mechanism to, to make this IMP, this AMP adenosine monophosphate, then inosine monophosphate, then guanosine monophosphate. I assume that all of you might be aware about these adenine, I mean purine and pyrimidines. Purines mean, purines and pyrimidines. I assume that you know about the genetic material DNA and RNA. I'm not entering over there. But the thing I wanted to point out over here that we have, we are going to generate our IMP inosine monophosphate and, xanthine monophosphate and guanine monophosphate, which are the basically part of the purine pathway. And this is actually is going on in the intestine, but parasite also sits into the intestine. Parasite also has to grow. So in order to grow, it needs its nutrient. That nutrient it takes from the host. So from the host is going to take this ADO. ADO gets converted to adenosine monophosphate. That gets converted into inosine monophosphate. This inosine monophosphate is the key intermediate inosine monophosphate, which basically gets converted into the guanosine monophosphate, which are the part of the DNA and the RNA. So from inosine monophosphate to xanthine monophosphate conversion is essential. And that essential happens through IMPDH, inosine monophosphate dehydrogenase enzyme. And this enzyme is located in parasite. This enzyme is also present in the human. You can see over here, IMPDH, inosine monophosphate dehydrogenase, that is essential to make the genetic material. So this parasite basically takes the material from human, AMP gets converted into ADO, ADO basically gets absorbed into the parasite, and then parasite is going to propagate, is going to make its DNA and RNA. And in this process, the IMPDH is an important enzyme which plays a key role. It's also important for the host as well. So Cryptosporidium Parvum, if we want to target this parasite, certainly we need to focus on key enzyme, which is inosine monophosphate dehydrogenase, called as in short IMPDH. Okay, so I just wanted to explain this cycle because it's very important. If you want to, target some drugs, say for example, if you think about what should I think for this pandemic, so you will certainly think of the key enzymes which are present in that particular organism. Whatever the key enzyme which plays an important role, there might be multiple enzymes over here, but the key enzyme is IMPDH, which is the rate limiting enzyme because it's going to control the synthesis of DNA and RNA. So you always like to block that key enzyme. Now you can correlate with this one with the current pandemic. If you look at the current pandemic situation, so the key enzyme, if you might have referred the current research which is going on to develop the drugs against this this virus, the pandemic virus. So you will see that main protease is one of the key enzyme which basically plays a very important role because that also is the rate limiting in the development of the virus. So there's a lot of literature is coming up to develop the MPRO, main protease of this virus to kill it. The analogy which I'm going to present in this talk is always, going to be more useful. So the essence which I'm going to deliver, which I'm presenting here, that is going to play a very important role. So you can apply for the, for the other pathogens as well. So if you look at what this IMP basically IMPDH is doing in this parasite, so it's basically taking this inosine monophosphate. I assume that you are aware about the structure of all these things, so I'm not talking into detail about that. So it basically takes this IMP and it converts into the XMP, which is xanthine monophosphate. So you can see here this H is being replaced with the oxygen. And that happens with help of the cofactor NAD, and that gets converted into NADH. So here again, the message I wanted to convey whenever you talk, if say for example, you want to target the main protease to kill this, this COVID-19, so you will think about which is the key, target which is responsible for the multiplication of this virus. So we will come to the main protein is an important target. Like here, the IMPDH is an important target. So the next thing which comes about, how that is playing a role in multiplication of that virus. Like here, the IMPDH is playing an important role in multiplication of this parasite. So you look into the mechanism. So mechanism explains a lot of the things. Okay, this is basically converting the IMP into XMP, and during this process, the cofactor plays an important role. And this cofactor is basically over here is the NAD, nicotinamide adenine dinucleotide, gets converted into NADH. So when you talk about the main protease of the COVID-19, you will see the cofactors are required. I will talk about the detail on IMPDH, and then you can correlate with this one. So this is can be a target which can give a lot of revenue if we successfully develop some of the drugs, because it's a rate limiting step. Now question comes, the nature of the pathogen is always very important before you think about targeting it. Like if you talk about the COVID-19, it's RNA virus, we know very well. But when you talk about the, so the very important thing is that nature is very important. Like Cryptosporidium Parvum is a parasite who is a prokaryotic in the nature. Okay, while there are several of the eukaryotic pathogens as well. So there are eukaryotic, probably you might have heard about of these eukaryotic pathogens like Plasmodium or like Trypanosoma. These are the very well-known eukaryotic pathogens. Cryptosporidium, Helicobacter, I'm not sure whether you might have heard about Helicobacter. It is a common which causes the, gastric acidity, and if long time it persists, it can cause the gastric cancer as well. So Helicobacter is again is a prokaryotic pathogen. And Campylobacter is another one. The reason is very important. If you want to target a prokaryotic pathogen versus the eukaryotic pathogen, your approach is going to be very different, because prokaryotic and eukaryotic pathogens, they have their nature is going to be very different. Um, so the IMPDH reaction, if you look at a little bit more carefully, so the what happens in this course of the reaction, this is I am discussing to make sure that if you think of a new target, if you want to engineer a drug, you need to understand, you should have a very strong understanding on the target. So target is IMPDH, selection of the target. First selection of the target is important, followed by understanding its mechanism, how it is doing this function. So if you think about, if you look at the mechanism of this enzyme, so enzyme mechanisms are quite common. So I'm putting in general. This can be applied to many of the cases. I mean, pretty much all of the enzyme, if you are working with the enzymes based drug development, or if you are aware about, if you're making a formulation which is, targeting a specific enzyme, so the mechanism of the enzymes are more or less common. There will be slightly differences. And that's the thing I'm going to present in next coming slides. And then I'll come to the drug engineering against that enzyme. So here, if you see at that most of the enzyme, you will see a nucleophile is always present, and that nucleophile may be of the from cysteine residue of the enzyme, or that may be of the some other. But cysteine is quite common, so it attacks onto cysteine. It's a nucleophile. It attacks onto IMP at the second position. This is the cofactor. This is a very important thing. You need to understand that in your enzyme mechanisms, you will always have a cofactor. So cofactor just sit nearby. It sits just nearby to this second position. As soon as it's going to attack, cofactor is going to grab a proton. It's going to grab a proton. It's going to come out from the pocket. So you have a pocket at the active site. That active site is accommodating your substrate. It is also accommodating the cofactor. It's interesting, accommodating both the things, the substrate as well as the cofactor. The nucleophile of the enzyme comes into, cofactor sits nearby, going to grab a proton, kicks out from the pocket, it forms an intermediate, water molecule enters into, it's going to kick off this, this covalent bond which formed through the attack of cysteine, and eventually you are going to get XMP, because this is a water molecule which is going to stick over there. I'm not again discussing the chemical reaction again in detail. If you like to, you can refer or we can discuss. But it's pretty straightforward reaction. So this is the way enzyme is going to catalyze this reaction. So if you look at little bit more detail, if you talk about the the target for COVID-19 and the IMPDH, there is an analogy. The reason I said that because majority of the target, if you see for the pathogens, they are quite relevant to each other. They're not much difference. Only the difference comes because of the pathogenicity. As I said that some pathogens, they are quite self-limiting, but many are not. Earlier also the the COVID came. I mean, as it's not like that this is the COVID is coming the first time, but earlier it used to be epidemic. It used to be epidemic localized into one area, but now it is a pandemic. It has been spread all over the world. That's a part of the major problem. And second is the pathogenicity. Whatever earlier the SARS coronaviruses which came into the world, they were not spread to all across the world. They were very limited to some specific countries, but this has spread to all over the world. That's why it's a problem. Second is the pathogenicity. This one is more pathogenic in comparison to the earlier viruses. That's the second fear. So if you talk about the why the more pathogenicity, that comes through the mechanism of this, the mechanism of the multiplication of the pathogens. Here I am talking with respect to a parasite. It can be applied to the to other pathogens as well. So if you look at the active site of this parasite of this enzyme, it's quite interesting. As a common phenomena which you will see in many of the enzyme, it is true. If you refer the recent papers which are coming up to which are coming up to explain the mechanism of the drugs which are being used in the clinic for the SARS COVID-19, you will see, probably you might have heard about remdesivir, and you might know about a few other drugs as well. Chloroquine, hydroxychloroquine, this has been given to the patients, and they are coming they are able to help to some extent, may not be to a great extent, but to some extent. And they have some certainly mechanism, and that mechanism is always because the pathogens, and this cryptosporidium is also a pathogen, so the mechanism is going to be quite common. So here interesting mechanism I wanted to explain, which is going to be applicable to many pathogens. So the important thing, I'm not sure whether you are aware about this phenomena, which is actually usually more present in the parasites. See, the active site is quite interesting. It has two places. One where substrates can basically sit into, another place where the cofactor sits into. So the substrate and cofactor, there are two places, one substrate, other the cofactor. The other important phenomena, this is the platform where these two are going to sit nearby to each other. And top of that, there is a loop. This is the flap. Top of that, there is a flap. The flap is going to close into and execute this reaction and eventually will form the product. So this is a common phenomena which is present in many of the pathogens, including the HIV protease. If you look at the enzyme mechanism of HIV protease, is the same more or less. It also has a loop, it has a flap. Loop has some specific amino acid where the substrate can sit, where the cofactor can sit. The loop has a specific amino acid where the substrate and cofactor can sit. Flap is going to have a certain specific amino acid. It gets closed once both are going to sit and going to execute the reaction, and eventually your product is going to get released. So loop and flap is a very common mechanism which is applicable to many of the pathogens. I'm taking it with respect to the IMPDH, but it's applicable to to many with respect to many of the enzymes which are present in the pathogen. So you can see here they both are sitting together close by. IMP gets converted into XMP. I'm putting a star, and NAD gets converted into NADH. It's going to come out, and now you can see that the flap is opening. So closing and opening phenomena of the flap is a very important. Flap has to sit inside. Flap has to come inside to execute the reaction. Once it is out, flap will be opening. Then flap is going to again close out. XMP star is going to, because now water molecule has to do the reaction. then is going to open up again, XMP is going to get released, and now it is ready to execute the next reaction. As you know very well, all these enzymes, their reactivity depends upon their kinetics, Some of the enzyme, they do this reaction in nanosecond. So it's going to is taking substrate very fastly and going to form the product, which actually we are unable to measure also. So I'm trying to explain over here, the pathogens are really very smart. They know how to propagate inside their host. Is very difficult to deal with for us. So other things. So this is something which I have explained about in general, that this is the mechanism which happens with respect to the IMPDH, and this is applicable to many pathogens. Similarly, the next thing which I'm putting here, the general structure of IMPDH. If you this again applicable to pretty much many pathogens, applicable to HIV protease and with the COVID-19 and earlier COVIDs as well. So very important thing that you need to look for the structure of that specific target. The target may be sometime monomeric in the nature, or it may be tetrameric or dimeric in the nature. I mean to say, it might have 1, 2, 3, 4, 4 lobes. It might have a single lobe, or it might have the 2 lobes. It depends upon the nature of the target. That will come to know from the X-ray crystal structure. If you recently look for, I'm sure that you know about protein data bank. Protein data bank is a place where you can find the crystal structures of the proteins. And you will see that based on the crystal structure, you will find out whether it is a monomeric, dimeric, or trimeric, or tetrameric. It depends upon the nature of the enzyme also. Okay, so sometimes what happens, the one loop is enough to convert the substrate into the product, but many times the 4 loops are required and they work in synergy. That's what happens with HIV protease. So this is true with IMPDH as well. Earlier it was not known, but now it is very clear that for the IMPDH, to execute the reaction, IMPDH is required at least 2 loops. Minimum 2 loops are required. So now when you talk about the little bit, look into the more into the PDV crystal structure, so very important phenomena, as I talked about in the earlier slides, you have a loop and then you have a flap. So the flap, as I said, is going to fold into the active site. I assume that you know about the active site. Active site or binding site is a place where the substrate used to bind in the enzyme. आप लोगों ने अगर enzyme का पढ़ा होगा, then probably you might be aware that in the enzyme we say with respect to any target, active site or binding site is a place where your molecule used to bind. Like if you want to make a new drug that's supposed to bind at a specific place on the target, that place is called as an active site, or you can say the binding site. Once it binds, it's going to produce the response. Certainly, binding site is the place where it should produce the response, maybe agonist or maybe antagonist. And sometimes it may not produce also, but it's a different category of the class of the drug molecules. So as I said, that there is a loop and there is a flap. So flap basically is going to fold in the NAD binding site. If you remember, I said that there are the 2 places. One is the IMP and other is the NAD. And this is your flap. That flap basically, sits into NAD site. So flap basically folds in the NAD binding site. Very important, very interesting point. Flap is folding in the NAD binding site, which is a cofactor binding site. Okay, is giving a very good insight that if I want to block this, this interconversion, I can actually work around where the flap is going to bind. So I mean to say, I can actually, if I want to make a drug against this pathogen, I have 2 places majorly. One where substrate binds. So I can make something which basically goes and binds there and does not allow to bind the substrate of this enzyme. One is possibility. Second possibility, I can make something which can basically bind at the cofactor site. Okay, the very important point which comes into picture, that the flap of this with this pathogen, it folds into the NAD, into the cofactor binding site. So I can think of targeting the cofactor binding site. आप लोगों ने, if you might have heard about, you want to make a formulation or a drug which basically might be a competitive inhibitor, which might be non-competitive inhibitor. I will discuss in the coming slides. So that comes from here. that where is your formulation is going to go and bind at the active site. And to prove that one is always challenging. And if you prove that, then you can always publish into the high journal and you can get a lot of funding. So very important phenomena. As we talked about that, there are the two places, one where the IMP substrate is sits, and other where the the NAD binding site is there. So the nicotinamide adenine dinucleotide is the NAD. If you look at the NAD, so this is an IMP binding site, and this is the nicotinamide adenine dinucleotide binding site. If you look at this conservative, so you will find that IMP site is 100%, is a similar. So it is actually similar in more or less in all of the pathogens where the IMPDH is present. See, IMPDH is enzyme which might be present in many of the pathogens. Is not necessary though it is present in the Cryptosporidium Parvum. It is present in the Bacillus. It is present in other of the pathogens. I will show the long list of the pathogens in which this enzyme is present. So whenever you are making a drug against, you might have heard about broad spectrum antibiotic and narrow spectrum antibiotics. So broad spectrum mean you are developing an antibiotic which can kill many of types of the bacteria, not limited to one. Like if you talk about the current pandemic, we are looking for very specific that should specifically kill the COVID-19. Okay, so we are not looking for a broad spectrum. We are looking for a very narrow spectrum drug at this time. But many times you look for the broad spectrum drug, which can basically, this is always ideal in making a drug for the, or engineering a drug against the pathogen. So in that situation, we always look for the sites which are either for behind or the highly similar or the highly divergent. So if you look at the NAD site, that's highly divergent, where the nicotinamide, this is the nicotinamide site where this nicotine ring is sitting. Okay. So this is highly divergent, and this is the highly conserved site. It's an interesting point. It's going to help a lot. So based on this one, this is something, the whole presentation I am doing is on, as I said, that is from my postdoctoral work. And currently also as a co-PI, I have one project against COVID-19 funded from SCRB. Recently got funded from SCRB pretty much a couple of months ago. So we are working on that. So if you talk about, so we have actually developed a lot of assays, and from there I'm putting into very short. We came up with the with the 1,000 of the compounds. And these compounds were screened through high throughput screening. I'm not talking about high throughput screening. And from there, we could came up with a compound which are, very selective against Cryptosporidium Parvum IMPDH. See, I said that IMPDH is present in human beings as well. So you want to make a molecule which should be more, acting more towards the Cryptosporidium, less towards the human. Okay. In generally, when you talk about the pathogen, drug discovery against the pathogen, you will always try to select which should be exclusively present in the pathogen. You will always try to avoid the target which is present in both the pathogen as well as its host. But many times you do not have any choice. You have to work around that, and it's always challenging. If you talk about the about COVID-19, you will see main protease is an important target because there is no counterpart present in the human beings. So if you can develop a drug against main protease of SARS coronaviruses, you can make a drug very easily. It's present exclusively into the pathogen, not into the human. But Cryptosporidium is always present in the human, required for our genetic material as well. So you like to have a compound, you like to have a drug which basically should work against only the Cryptosporidium. So a lot of assays were done, and we came up with the sum of the molecules through high throughput screening. So here are the sum of the molecules which I'm putting up. As I will progress, it will going to be more and more clear to you, how to engineer against a pathogen. So this was done through the high throughput screening, and from there we came up with these compounds, and they are, very active against Cryptosporidium IMPDH, not against the human IMPDH. The very important point, if you look at these structures, because I'm talking with respect to the, the engineering of a drug. So if you very important, if you look at the structure of these drugs, they are very structurally divergent. You have a triazine ring here, you have a amide bond with the aromatic feature, and then you have a very different structure. So structural diversity is quite a bit into these. And many times you see you have a molecule which is a urea derivative, which bears basically an oxime over there. So structural diversity is quite a bit over there. Okay, I'm not actually talking into the detail because I assume that that will be more towards the chemistry side. So I'm not discussing about there. If you have any question, you can ask me. We can discuss on that. So once you have, so here the engineering principle comes into, so once you have some molecule, you can see this molecule is is not, when you talk about the a drug molecule, you always like to have a drug molecule which should be more potent and should have the least toxicity. So if you see this compound, this has not very potent, IC50 is in micromolar. So you want to always develop a molecule where you can reduce this IC50. So in the important principle, when you see from the engineering principle comes over here. So when you see this one has very low activity, you want to convert into something which should have IC50. Now you can see in these compounds in the nanomolar. So from micromolar to all the way you can go to the nanomolar. So you are increasing the activity by 1,000 times. It's really interesting. So how you engineer that kind of the things. So first think what the basic template you have. From that template, you will always try to keep those features which are essential for the activity, which are really required to bind at the active site. I said about the NAD binding site is there. Okay, so some of the features you will always like to keep on, but many features you will modify. So if you look at this, aromatic feature was not changed, it was kept intact in the development of this whole molecule. Okay, the another important point comes that this carbon, if you look at in terms of the chemistry, you know very well, this is a chiral carbon. So you can basically, initially this is a racemic one, you can make it a chiral one. So now this has a specific orientation. So in this molecule, this can be go up, this can be down, but in this one, this is always up. This is down, sorry, always down. And over here, you have changed this feature with you have added one more phenyl ring into this one. So by doing all these structural modification, you can basically change the activity. You can engineer a drug which is going to be more potent towards the pathogen, towards the parasite, and you can increase the activity by more than 1,000-fold. This is small principle I'm talking about. I'll talk about in more detail. This is another series of the compound which we already had, which we identified through high throughput screening, but the activity was not very high. So then we can actually, you can see here, the activity of this compound is 300 nanomolar. But when we have done these modifications, it came to 1 nanomolar, which is a significant change. And all these we have done by keeping in the mind the features which are really necessary. We'll keep those features, but we will actually play around the feature which can be modified. Like here, you can do a lot of modifications. So you have done a lot of modifications. We have done a lot of modifications and came up the compound which has very low IC50. I assume that you know about IC50. It's the inhibition constant, 50 percentage. So it indicates whether the compound is killing the enzyme or not. So this is a very important principle which we need to learn. Here we are talking about the IC50. So my mean to say, all of these study has been done in vitro. So this is an enzyme-based study. So we have not seen so far whether this molecule can enter into the parasite or not. We are just indicating that yes, these molecules can inhibit the enzyme activity. I mean to say it can inhibit the conversion of XMP, IMP to the XMP. That it can do outside the parasite in the well plate. But in the parasite, whether it's feasible or not, for that we have to do other experiments. But this is the first thing which you will do if you want to, or if you have a formulation. So for the very first thing, you will actually test the formulation in vitro to find out whether that formulation is basically acting against the specific enzyme or not. You might be taking a standard drug which is available in the market for some of the disease, and then you are developing a formulation to address some of the issue. But make sure that you have to retain this, the property of that original drug in terms of its target. Like if it is drug is targeting the drug is targeting the IMPDH, so that IMPDH activity should be retained. So that's the first thing we do. So here we can see that, we can improve the activity by, doing all these things. Okay. Now, when you talk about these all inhibitors, when we looked into this, so that's another aspect which comes into the picture is the X-ray crystallography. So what we have done here, that we have the molecules which we developed and where we have improved this, the enzyme inhibition properties to a significant extent. Those molecules were crystallized. And the X-ray crystallography is very much is giving a lot of information. And what it is indicating, that these inhibitors, they are basically mimicking the NAD plus. It's mean they are more occupying the nicotinamide adenine dinucleoside, NAD site. So if you look at the enzyme, I'll show in the coming slides as well again. So these are more towards the NAD site. They are binding onto the NAD site. This is very interesting, along with occupying the IMP site. So they are binding to the both of the places. That's the first of the interesting point. The second interesting point, that this NAD binding site is a different, is located at different place. That I'll talk about when I talk about the bacteria. I'll talk about that site. So here, if you look at this, that's the thing I wanted to mention over here. In earlier, one of my earlier slides, if you remember, let me go back and bring it over here again. So I said that IMPDH is a square planar tetramer. So 4 units are there in the enzyme. So very important and interesting point, because that's something is very important to know if you really want to engineer the drug in a correct manner. So the very important point which comes into that, these inhibitors, they binds in the adjacent subunit. So there are the 4 sites are there in the enzyme. Enzyme is tetrameric in the nature. And these inhibitors, they are binding, some part is binding into the one pocket and some part is going into the another pocket. Some is over here and some is located over here. So you can see the different colors. They are indicating the different, different loops. There's the one is a tetramer. So I'm just showing the 2 units over here. This is the 1 unit and this is another unit of the enzyme. And you can see here these compounds, they binds on the both of the places. They are binding into the 1 unit and then they are migrating into the another unit. That's a very important principle, that they are migrating into the another unit. They are blocking the 2 sides of the target. It's mean that they can be probably more effective in comparison to the drugs which are just binding into the single site. So again, as you can see over here, this is the this is the one of the molecule with an X-ray crystallized over here, and is important is that how it is binding. So it is occupying this IMP site, and this also occupying the NAD site. So both the sites is going to occupy, and it's binding into the 2 pockets. One of the 1 unit, and other is going to the another unit. Then this we have actually shown with more or less all of these molecules. Their binding pattern is very unique. They binds into the adjacent pocket, and that's can you give you the another principle in the designing and engineering of these molecules. You see another molecule also is binding the same pattern. So their binding pattern is the same. So because their binding pattern is the same, it is going to give you another information, that which amino acids are there with which this is going to interact, and that's how it is going to kill your enzyme. So all the pattern again with this molecule also. So with as I have shown in the earlier slide, that the compounds, they are structurally divergent. Okay, so here you can see they are structurally divergent. And the important principle what we have observed here, that in all these structural diversity, but their binding mode is the same. See, all these binding mode is the same. You will see these 2 residues are there. 2 residues are there. So here these 2 residues, here these 2 residues, here these 2 residues. Okay, here these 2 residues, and this is a third residue. This means that the binding mode pattern is giving you that which amino acids are really playing a very important role with respect to all these molecules. So 1, 2, 3. Okay, these are the quite interesting where all these molecules are binding. Okay, this can be utilized in the designing of, in engineering of your molecules. So here is important point comes. Okay, so these residues are what these residues are. These are the three residues: one alanine 165, another tyrosine, third is the glutamic acid 329. So these are the three residues: glutamic acid, alanine, tyrosine. On all these you will see glutamic acid, alanine, tyrosine, glutamic acid, alanine, tyrosine, glutamic acid, alanine, tyrosine. Everywhere these residues are there. Okay, so this is the something which can actually explain you what you should do if you want to further improve the activity of these compounds. So you have a lot of information now. One information, that their binding is very unique. They bind into the two sites. Second, and the second site where they are binding, these amino acids are playing a critical role. So we can actually play around these to do further modifications and develop the molecule. So here I am trying to explain you in general what we have studied in our undergraduate in detail about the enzyme inhibition. So you know very well, as I have explained, that this is an as IMPs, this is your enzyme where this IMP is going to sit. NAD cofactor will come into the picture, hydride transfer will take place, and then NADH will be departing from here. You have the enzyme with XMP open state, then it is going to close state where your water molecule will come and do the slow hydrolysis, and eventually product is going to get released. Now it depends upon the compound which we have developed. As I said, that they are killing the enzyme which we have shown through these in vitro assays. The assays were not done with the parasite. Okay, it's very important. So here you can see depends upon the nature where they are blocking. Based on that, you can name them like competitive inhibition. Competitive mean they are going to compete with the with the binding site. So here ideally NAD is supposed to come, but NAD is not coming. Your inhibitor is coming. So as a competitive inhibitor is quite familiar. All of you, I'm almost positive. This is an uncompetitive inhibition where already NAD got departed, but now it is not allowing the water molecule to enter and execute the reaction. Loop is not, it still remains into the open conformation. So it's an uncompetitive inhibition. Non-competitive inhibition. Non-competitive mean it's actually is going to compete that site, but NAD has already, occupied that one. So it's not sitting onto exactly on the same site, but sitting onto slightly the another site. It's a non-competitive inhibition. So far story looks quite good and interesting, but we still are lacking with respect to many of the things. As I said, we do not have any evidence to show whether this can kill the parasite or not, because that's always very difficult. The reason is that that's a problem with, if you talk about the COVID-19, you can give a molecule, but testing against the COVID-19 is difficult. Where you are going to grow the virus, it's so hazardous. It can infect the person who is going to do the all these things. So doing these testing the drugs against the pathogen is always challenging. The reason is that continuous culture is very difficult. Okay, and if you culture also, you cannot separate the parasite from the host. So how you are going to do that? So in COVID you can separate it, but you cannot culture it. Once you culture, it can infect that can cause the problem. And there is no genetic tool. Probably you might have some genetic tool. I mean, there are genetic tools as well to study the mechanism of the actions, but there is no genetic tool. There's no animal model. The same true is with the majority of the pathogens. The problem with all the pathogens, animal models and testing against the animal model is very difficult. You talk about the anti-TB drugs or any of the pathogen. With all these, you have these are the common challenges which you will face. So there is a way to work around. Certainly, as I said, you cannot do this thing in the parasite, because parasite you cannot separate from the host. Once you separate, it's going to die. So what you can do, you can select another parasite which is quite similar. So that will give you very strong evidence, but not 100% proof. Okay. So the other organism was selected as Toxoplasma. It's okay to grow. You can test the drugs. So that's why this was the pathogen was selected against this. You can actually do the study outside the human beings. You can grow this one and you can test the compounds. It has the structurally, these enzymes are very much similar. So that's another evidence. So the So if we have the problem, there is always way to come around. Cryptosporidium, the parasite you cannot grow outside. So you took something, you actually selected another parasite, which basically has some, to a significant extent, has similarity with the pathogens. Again, story is not complete. Okay, you are taking not the real parasite, you are testing against the other parasite. It's not necessary though that once you give to the to patients, it's going to work. You might so this is kind of the red flag, you say, in general, against the when you talk about the all these drug engineering. So the parasite, this parasite, new parasite, can be can be stable, can be easily, you can easily express the gene fluorescent you can use, and you can always monitor whether your drug is working or not. It is going to reduce this green fluorescence. It's mean that your molecule is perfectly okay. Okay, the drug is against this parasite as antiparasitic activity is hard. Enzyme inhibition is not easy. So nitoxanide is a drug which is available in the market, is not a good drug. The reason is that because it has a nitro group, it causes the cardiotoxicity. Is not a good drug for the children specifically, and for the elder people as well. It do not behaves like a like a drug. The one of the possible reason is the nitro group, which is reported in the literature against the limitation of the nitoxanide. So what you should do, so you should try to develop a animal model where you can test it. So animal model was developed to test the compound which we have developed in the lab against Cryptosporidium. See, I'm talking about a parasite so far, Cryptosporidium Parvum. And we have tested this molecule, and I'm not again talking in detail, just trying to put it over here that it is really very effective when you do administer to the animals 3 times a day. It is potent than paramomycin, which is the drug in the market for to kill the Cryptosporidium. It is very effective at the lower doses. And when you do the single time, then it's not more effective, but when you do administer 3 times, that is more effective. So that's why if you heard about the recent vaccines should be administered at least 2 times, 3 times. Not a single time is not effective. The reason is that it depends upon the efficacy of the molecule. Sometimes they are not very effective in a single dose, so you have to divide the dose into the 3. You're giving still 250, but you are dividing into the 3s to achieve your activity. It depends upon the nature of the pathogen. So what we have found that instead of single administration, if you do a 3 times administration, the same dose is going to be more effective in comparison to a single time giving the highest dose. So next comes into, okay, so we know that IC50 is of this enzyme which I have talked about, and then the pharmacokinetic studies were done. I'm not talking about in detail. What the interesting point over here was that the concentration, which generally we say is that more the Cmax you have, more the biological activity you are going to have. That was not turned out to be true here. The compounds which had the less concentration, Cmax was the less, still they were very active. And there was some reason behind that, why this unusual phenomena happened with the antiparasitic drugs. So the plasma exposure was, AUC was calculated. You can see even the low AUC, then these low AUC compounds are, these are the active one. And there was some specific reason for these, molecules. So I'm not talking about why these are the why these even though AUC is less, again even still the compound is active because there is a because in what happens that, just briefly to tell you about you, so you will not be confused. The reason is that this molecule basically can undergo the enterohepatic recycling. So is either you are administering one time, but is undergoing again the enterohepatic recycling, keep coming, going, coming, going, coming, going. It is not excreted from the body, so it's going to stay for a longer time in the intestine and it's going to produce the more effect. So AUC is the less, but again is keep going into the cycle. So that's more effective. Okay, this so far the story was against the parasite. Now I'm entering against the pathogen, which is the antibacteria. Okay, so here is the question comes about the engineering of the drug from parasite to pathogen. You might have heard about the drug repurposing. This drug repurposing is slightly different comparison to the comparison to the points which I'm going to discuss in the coming slides. So the molecules which we have developed, they are active against Cryptosporidium Parvum, but once you test these molecules against the Bacillus, this is the one of the Bacillus anthracis is one of the pathogenic organism. which has been actually caused so much of the problem for the human being. So Bacillus anthracis, if you look at the IC50 against these, this is very low. If you look about the Cryptosporidium Parvum, even the target is the same. It's mean that something is different with this bacteria. When you talk about the MIC, I assume that आप लोगों को पता होगा IC50 is a different issue. As I said, IC50 का मतलब है कि आपने कोई bacteria को, you have not utilized any kind of the pathogen. You are just taking the enzyme. But when you talk about the MIC, it's mean that you have the bacteria, you have cultured the pathogen, and then you are putting your molecule. So when you talk about the MIC, you can see the MIC is very high. This compound is quite active against Cryptosporidium parvum, but against the bacteria, it is not effective. So why it is not effective? So certainly there should be a different reason. The reason comes because the parasite has a different kind of the barriers which is actually required for a molecule to cross and kill the parasite. But bacteria has a different kind of the barrier. I think all of you know, if you talk about the viruses, they have a different kind of the barrier which is required for a molecule to enter and kill the pathogen. So here is the list of the IMPDH. There's so many bacteria you can see. They all are carrying inosine monophosphate dehydrogenase enzyme. So certainly the molecules which we have proposed, these should kill all of these, but that's not the practically true. The reason is that their environment, the outer coating of these pathogens is very different from one pathogen to another pathogen, and that makes the more challenging of the drug engineering. From when you actually talk about a drug which is already known for one of the parasite, you want to convert that it should act against the other pathogen, it's going to be more challenging. Okay, so what's the uniqueness which can be utilized? Certainly always challenges are there, but there should be some uniqueness. So uniqueness is that the conservation of the inhibitor binding site, that's the very uniqueness. Majority of these pathogens, they are harboring these 3 amino acid residues. If you remember, if I talked about those 3 amino acids are still present in these pathogens. So certainly you can modify these molecules to kill these to pathogens. So when you talk about the bacterial, now I'm talking about the bacterial IMPDH. Earlier I talked about the parasite Cryptosporidium IMPDH. So when you talk about the bacterial IMPDH, very beautiful thing you look about. The cofactor, which is basically NAD, if you remember, this cofactor binds in a different conformation. It is very compacted in comparison to the Cryptosporidium IMPDH cofactor binding. In Cryptosporidium, it binds into the open. It's quite large. But when you talk about the bacterial IMPDH, is cofactor binding is very compact, and that's how the challenge comes into that these compounds which we have actually tested for the parasite, they are not active against the pathogen. So then what we have done, we have developed a lot of the molecules and try to plot a graph. What you can see over here, here is the IC50 against the Cryptosporidium IMPDH. Here is the Bacillus anthracis IMPDH. Both are the same enzyme IMPDH, but you see here these compounds which are against the Cryptosporidium, active against Cryptosporidium, but they are not very active against the Bacillus IMPDH. The reason is that cofactor binding is very different. So that's why these compounds are not very active. In terms of IC50, again, you need to understand here, I am talking about the IC50, where we are not actually involving any kind of the pathogens. We are talking about just enzyme of the bacteria. We're taking these IMPDH from the bacteria. It's commercially available. You can make in the lab, usually make in the lab. And then you put your compound.
Hello, sir. So can you unmute?
Sir, you are muted. For some reason, your voice is not coming, sir.
Hello, sir.
Hello?
It's coming?
Yeah, now it is coming, sir.
Okay. You always stop me if some problem is there. So now if you look at this graph over here, so if you look at now I'm talking about the, MIC, it's mean we have taken really the bacteria. So if you talk about this, IC50 of these compounds against the Bacillus, IC50 is mean where we are not taking any bacteria. versus the MIC where we have taken the bacteria. What you see over here, there is no much correlation between the IC50, although the IC50 is very less, but MIC is very high. It's mean, what is it indicating? That these compounds are killing the enzyme, not all, but few. They're able to kill the enzyme at low concentration, but their MICs are very high. You see the 100 micromolar is going beyond 10 micro. All majority compounds, the MIC you see, and IC50 you see, they are sitting more than the 1 micromolar, or with respect to this one is more than, they're always high, on the higher end. It's mean these compounds they are not basically, these are able to kill the Cryptosporidium, but they are not able to kill the Bacillus IMPDH, and they are not able to, kill the pathogenic bacteria. So MICs are very, very high. All are sitting on the, above the, I should say 5 micromolar. This is not a very good MIC. So what can be done over here? Oops. I'm not moving next. Okay, yeah. So here is the principle of the engineering comes into. If you talk about all these compounds which are acting against the Cryptosporidium, their C-log-P value is an important parameter. Another is the TPSA, which is also important parameter. These are around 4 to 67, okay? The compounds which are killing the Cryptosporidium parasite, their C-log-P and TPSA are around this range. C-log-P is indicating how much compound is in between oil is to water. I'm not talking about in detail, but C-log-P is oil is to water partition coefficient. This is an important coefficient which indicates whether compound can enter and cross the biological membranes or not. Probably you might have heard about class 1, class 4 drugs, okay? Different classes of the drugs, BCS classification. So some drugs are water soluble, some drugs are not water soluble. So C-log-P is having some correlation over there, okay? So C-log-P is in the around the range of 3.5 to 83 for all the drugs which are actually active against the parasite. Now, interesting point. The drugs which are available in the market to kill the gram-positive, I talked about Bacillus, right? It's a gram-positive bacteria. E. coli is a gram-negative bacteria. So the drugs which are available in the market, their C-log-P is very low, it's around 2. The TPSA is very high, but when you talk about the gram-negative, is further very low, is negative side. TPSA is around, over than 150 Armstrong. So it's mean that these compounds which we have developed, they are not entering into the bacteria because these values are not optimal. Okay, so the goal comes that you need to actually, change the structure of these molecules. You have to engineer these drugs in such a way that these compounds which are killing the Cryptosporidium, they should also kill the bacteria. How you do that? So you are going to change the C-log-P and TPSA through the chemical modification. So important principle comes over here. If you want to, design a specific and engineer the specific drugs against one pathogen to another pathogen, you need to look for its properties. Okay, so cofactor, as I already explained, that it always binds into the nearby adjacent subunit. So that's a very important point. Adjacent subunit, cofactor mean NAD. It binds into the adjacent subunits in the all pathogens. Okay, and the important point over here, how we do it. So if you look at the crystal structure of Bacillus IMPDH, the interesting point you will look for that the inhibitor binding pocket. As I said, that the cofactor binds into a compact conformation. And the binding site of the cofactor is located on the second loop. So 2 loops are involved in the overall process. So this cofactor binding site is quite larger in Bacillus IMPDH. Okay, so this cofactor binding site is the larger. That's the point we got. We also know that what we need to change these properties. Okay, so now let's actually engineer the drugs with the existing molecules and came up with a new molecule. So what we have done, I'm not sure, probably you might not be remembering. So let me actually go back to one of the previous slide, the structure I should have pulled up over here. So you can see over here. So this is the one of the molecule. Okay, you see here, you have a chloro, you have a nitro. Okay, we know that this site is binding where it's binding onto the cofactor binding site, which is quite large in the nature. Okay, we need to reduce the C-log-P, we need to improve the TPSA. Where we will be doing these modifications, we will be doing modifications over here because this is the place which is the larger in the bacteria. Okay, so it can basically accommodate the groups, larger groups. Okay, so larger group it can accommodate. So that's what we have done in the engineering of this one. So you can see here, This is the same part. I mean, the other chain was removed, but here you see here we have is the instead of nitro, we are putting a bigger group over here. We have also, so this is the thiazole we called, and this is called as a pyrazole. We are putting a bigger group over here. So as soon as you put a bigger group, you can see over here, the MIC values of these compounds is drastically goes, down. So these are the very active against the, against the pathogen. IC50 values are also decent. It's not very bad, around 35 and 15 nanomolar. And MIC, it means they are able to kill the pathogen also. And the MICs are around 1 and around the 1 micromolar, which is really very low. Earlier we had MICs more than 50. So it's mean that these modifications are working. So the principle we have learned over here that we should, just a minute, that we have to select the specific place where we have to do the modification that we have selected through the crystallographic. Then we have selected what modifications should be done. Modifications should be such that it can change the CLOCP and the TPSA. So by that way, we could actually come up with the molecule which basically, so this is applicable to pretty much all the pathogens. So now what is happening, what this MIC is indicating now? It is indicating that now the compound is able to enter into the bacteria, because it is killing the bacteria in very low concentration. Earlier compounds were not able to enter into the bacteria. That could be the one of the reasons that they were not effective and they were not killing the bacteria, and their MICs were very high. And these compounds can enter into this one. We have now done these modifications. Here you can see the TPSA, the CLOCP values, and their MICs. Okay. The second modification, if you look at this structure, the next structure which I'm going to show, so is going to be the same, except that instead of this methyl, you will see NH2 group. Okay, NH2 group. So as soon as you replace the CH3 with the NH2, you are going to change the TPSA to a significant extent. You can see here 91 to, you are going to 99, and so it's the same structure we should compare. If you look at this structure is the 91, here is the 117. There's a drastic change in TPSA, and you can also change the CLOCP. Earlier it was 4.47, now it is 3.36. So we have changed these things to a significant extent, and we have seen that MIC is actually going down to a drastic extent. You can see here the experiment was done in presence of guanine, and in the absence and in the presence. So guanine is something which basically when you give to the bacteria, bacteria can utilize it, and bacteria can make their own IMP. So you do not actually, they do not need to take the IMP from the human beings. So once you give the guanine, the MIC is increasing. It is indicating the compounds are on target. So it's another thing which you have to prove with your compounds. So this is a phylogenetic tree. Will be actually finishing up another 5 minutes. So the phylogenetic tree, if you look about, so you always look about this, closeness is indicating that, how close these are. So this is Baumani, this is staph. One is a gram positive and gram negative, but they are quite close to each other. So it's mean the compound which we have developed, they should also be active against BM. Here is the assay staph and the BM, and they are very similar in the function, yet they are the divergent in the sequence. So this is something I am trying to show over here, that the compounds which we have developed against the staph, they should also be against the Baumani, with another pathogen bacteria. The same thing is over here. This Bacillus is also similar. So because of the sequence similarity, it is expected these compounds will also be active against the pathogens. So flap plays a very important role that controls the overall inhibitory affinity. So it's a very important take-home message. So the take-home message from so far is that if intestinal exposure is the more, it can always increase the efficacy when you talk about the Cryptosporidium Parvum. Other important thing which you always talk about and which you always keep in the mind when you talk about the antibiotic drug development, because it should not perturb your normal gut microbiota. That's why I said developing the drugs against pathogens is challenging. You can see the current situation also. It's a very challenging, right? So because it can perturb the normal microbiota, that can give another challenge. So that also you have to do. One of the compound I'm presenting here, which we have developed, that was actually disturbing the gut microbiota. So we have to skip that compound from the study. or need to come up with other approach. So with that, I will be I'm finishing it up, and I should certainly acknowledge my PhD supervisor, Professor Alok Datta, and the all presentation which I have given here is from my postdoc work, which actually I did with Liz Hedstrom and Greg Cuny at Brandeis University and at Houston University. So we have a couple of projects, and one of the project also, as I said, is from SCRB on COVID-19 as a co-PI, which I've not indicated over here. So with this, I like to thank you all of you. Thank you very much. If you have questions, we can pick it. We can take a few of questions.
Thank you, sir. Dear delegates, if you have any questions, feel free to ask. You can also put your questions in chat box, or you can ask directly.
Yeah, you can ask directly, and if you can always take my email from madam, and you can communicate me through the email any point.
Yes, sir.
So if you have any question, please feel free to ask. We can discuss a couple of questions.
You can put your questions in chat box also. We will read for you, sir, if there are any questions.
Sure.
Yeah.
Sure. Thank you.
Yeah, I think delegates have understood the topic thoroughly. Yeah, that is very good. Yeah.
Anyway, fine, good. So please feel free to contact me. You can get my email address and stay in touch with you.
Yes, sir. Yeah, dear delegates, if you want to ask questions, sir, or communicate in future, you can take sir email ID and you can contact sir. Now, okay, we have come to the end of this today's session.
Okay, sounds good.
I thank you very much, sir, on my personal behalf and KLE's College of Pharmacy for your talk on drug engineering for pathogens. Very informative talk it was, and it helped our post-graduation students also, as well as all the delegates. We once again thank you very much, sir. Thank you.
Thank you very much. Another meeting is started. Thank you.
Yes,
I need to join that. Okay, thank you very much.
Sir. Thank you. Here is one announcement. We expect you people to be interactive. Okay, at least one or two questions you should ask. Okay, see, so many eminent speakers we invite, and if you don't interact, it doesn't look good. So if you cannot ask directly, you can put your questions in the chat box. So that is that much I think we can expect from you all. And please be on time, all of you. Log on, okay, on time. Don't log in late and all, because it is very embarrassing to make speakers to wait. Speakers, they join 5 minutes, 10 minutes early, and we have to keep on waiting till the some specific number of delegates to join in. So that should not be there. We are telling this again and again repeatedly. We are putting in WhatsApp messages also. So please maintain the timings properly. Okay, whenever the scheduled timings are there, please join in. Okay. Yeah, with this, I thank all the delegates, and we will meet tomorrow with the next session. Thank you, one and all.